Okay, let's talk about safety today. What I thought we would talk about is introduce our sheep studies. And the reason is because I want to tell you a little bit of background as to like where what is the complete picture for the safety studies? Like what did the sheep undergo before we looked at the safety aspects? But also because just like last time, I've been feeling kind of bad that we're spending all this time talking about the physics and not really kind of getting into something fun, like some actual neuromodulation. And so let's introduce our sheep studies so the students can actually see some like real experiments. And then there's comments and some other people's work as well. And then we'll talk about the safety study, ours as well as a few others as well. And then just some comments at the end. So this lecture should be short because I think I went really long on the last one. So
yeah, we did. Oh, Kim, before you start, why the sheep?
Why did we pick the sheep?
Yeah, just to get everyone in the mindset here.
Yeah, well, I'll talk about that as well. There's a couple of different reasons for that. One of them is that sheep have a relatively thin and homogeneous skull. And so since we weren't going to do any phase aberration correction, just because that was a lot to sort of organize, since it was homogeneous, we expected to get a reasonable focal spot. And then I'll show you some pictures of the focal spot with MRRFE. The other reason was that we wanted to do the safety study in sheep because we needed to redo the Sung Shik Yu's safety study in sheep. So I'll mention that again
in one of the slides.
Yeah. Okay, let's get started. All right, so we wanted to do large animal deep brain stimulation, and we wanted to do deep brain stimulation, so we wanted to pick a large animal model because you could argue that there is no part of the brain in the mouse which is actually deep. And so we picked the sheep model, and we picked the sonication of the LGN because this early paper that, again, Keith showed this in the first lecture from the Fry brothers, where they're sonicating the LGN and see the visual evoked potential, and it gets diminished and then it comes back. And so we were pretty sure that it was going to work, high probability of having an effect. And we wanted to go back and do a more thorough study because this is really almost the sum total of their methods in that sort of one column article in Science. Well, they said they sonicated the lateral geniculate nucleus of the cat. They also said they took the skull off the cat. They recorded the evoked potentials, and that was about it. So they didn't have any information on what their intensities were or the timing or really anything. So we wanted to do a little bit more thorough study.
Mm-hmm. It's not only the methods, but I think that figure is the entirety of the data from that paper, which is amazing, but it's not enough to really go off of.
Right. Okay, so this is our setup. We had a transducer, and we're focusing in on the LGN on one side of the brain. So we have LED flashing lights on both eyes. The optic nerve comes in, it crosses here at the optic chiasm. Well, some of it will cross and some will go to the same side. And then through the LGN, which is a relay point here, and back to primary visual cortex. We had electrodes over the primary visual cortex, and we had some that were further up in the front part of the brain. Okay, so a little bit more about our experimental setup. This is the transducer that we use. This is a flat transducer. It's an array. It has 1,000 elements. Right now, it has a membrane, and the membrane is filled with cooled circulating water. And so that keeps the elements cool. It also provides coupling. And being a phased array, then we could steer the beam as we wanted. So what we did was we attached that transducer to the top of the MRI head coil. And then we placed the sheep underneath the transducer and kind of snug up against that membrane so that he would be well coupled. You can see the flashing lights on his eyes. So as the sheep was up against the membrane, when they had a kind of a nice flat homogeneous part of the skull, as you can see here, this is the CT. And we did have the electrodes placed on his head. Then we put him in the MRI, and we did 2 sets of images that were important. The first one with the MRRFE. That was to make sure that we got a picture of the focal spot so we can make sure the equipment was working, and also to make sure that the beam was reasonable. And it always gave us a reasonable beam. It never looked like it was too distorted.
And then we know that it was targeting the LGN. Were you just using neuroanatomical landmarks and sort of guessing?
So then we used a T2 weighted 3D cube sequence such as this one, and then found the LGN from that.
Okay.
Okay, so, and I mentioned the MRRFE last time, so I guess I don't really need to get into it again. It did give us a nice picture of the focal spot, as you can see here. And okay, so then once we've done our imaging and we knew where the location was for the LGN, and then we also had picked out some control locations that were about 1.5 centimeters away from the LGN. Then we started flashing lights, and while we're flashing lights, we were allowing the anesthesia to lighten up. So we're collecting data from the EEG, we're looking at the visually evoked potential. We don't see it until the anesthesia lightens up enough, and then we start to see it, and then now we're collecting our baseline data. From then on, we maintain the heart rate and the temperature to be about the same throughout the rest of the experiment. The anesthesia that we had, which was initially tilazol, was sort of wearing off. And so we maintained with isoflurane, usually at about 0.3%, although I was monitoring the end tidal percentage in order to maintain a constant percent. All right, so we had 20 minutes where we sonicated LGN, and then another 20 minutes, and then we had 2 to 3 20-minute periods where we were sonicating a control location. And then during all of that, we're collecting data. We have interleaved 4 different stimuli. So the first stimulus period of 15 seconds was light only to those LEDs. The second 15 seconds was ultrasound only, then ultrasound and light, and then no stimulus. And the results I'm going to show today were from the light only stimulus. That being said, what we'll see is very different than it was during the baseline, which was light only, because it was done at the same time that there was another period where there was ultrasound. So we're looking at the effect of having ultrasound in an adjacent 15-second time period throughout that 20 minutes. So it wasn't due to the kind of a direct response from the ultrasound, but the effect of the ultrasound on the brain and how that affected the light only visually evoked potential. Okay, so those electrodes are on the back of the head, and we're measuring from the electrodes. And then what we are looking at are traces that look something like this. So the one where we're sonicating the LGN is in blue. And then when we're off target somewhere else in a control location, then we get the black. And so there's these 2 different peaks that we're looking at. So this is 70 milliseconds after the stimulus and then 100 milliseconds after the stimulus. And those are kind of known peaks for us to look at. And then we're looking at the difference between them. And you can see that that difference is much bigger here in this individual trace than it is when we were sonicating. Now, that's just one example. And then looking across 6 animals, and then we had also 5 controls that didn't have any sonication to the LGN. And then what we see is that the visually evoked potential, so it's the peak-to-peak measurement right here, is decreased from baseline when we are sonicating at the LGN location for the first 20 minutes period, the second 20-minute period, and then it came back again when we were sonicating the control locations. As you can see there.
Each one of those data points is 20 minutes apart, essentially?
Yeah.
Okay.
So we didn't have great temporal sampling because the temporal sampling was 20 minutes. But that, that is a limitation. So, whether or not this might have shown that there was a period before it came straight back is just not something that this data was telling us because of our temporal sampling.
Mm-hmm.
So that's really it. Just the fact that this was showing us that there was a suppression of the visually evoked potential. And, that's exactly what Fry saw. So it was great. We saw what he saw. It gives us a little bit more information. We were able to see that it came back again in the control at that period. We were able to measure the statistics, more or less what he saw. We were able also to put the skulls in the scan tank and measure the transmission efficiency so that then we could go back and estimate what was the ISPTA that we had actually used. And then you see that the peak-to-peak amplitude is related to the ISPTA. So, the timing that we used for all of the animals was the same. So you could easily just have changed this to ISPPA as well, because since the timing was the same, the ISPPA would be higher when the ISPTA is higher.
So Kim, can you tell us why you didn't sample higher, after the stimulation, why you didn't continue sampling? Because it's as easy as flashing a light and then measuring the electrical response, right?
So we measured that for an hour after we finished sonicating the LGN. Is that what you're asking? Why we didn't go longer than an hour?
No, I'm sort of asking. So from this, it's either on or off. The LGN is either completely suppressed or it's not, and the in between could be seconds, it could be minutes, it could be 20 minutes, right?
Yeah.
We just don't know.
Well,
And
so
not sample throughout the entire period, right?
So each 20-minute period yielded 300 trials of light only, and we used all of those for averaging the signal in order to add it.
I see. Okay.
So it's sort of a signal to noise question.
Right.
And so several times, one thing we could do is we could look back and say, well, do we have enough signal to noise if we looked at the first 150 measurements and the second 150 measurements in order to double our temporal resolution?
Mm-hmm.
Yeah.
Yeah, I'd be curious if you resampled the data and did like some sort of a smoothed average, because these are, it's a pretty big difference. So you might be able to see something.
Yeah. Okay, so it's very similar to what Fry saw, and it was also similar to what Della Piazza saw. And so he had done something similar where he had a median nerve stimulation, and then he looked at evoked potentials in the brain. And he also saw that evoked potentials decreased when they sonicated. So very similar results. Okay, so, I think this is nice because it's very straightforward and easy to understand. There's something else that we did that was kind of fun. Last lecture, I talked a lot about the mouse experiments, and we really tried very hard to find some lateralization in the mouse. So if we sonicated on the right side of the brain, would we see the left forelimbs twitch? And vice versa. If we sonicate on the left side, we would see the right only twitch, but not the left ones. And we never saw that kind of lateralization. It was always both forelimbs. So in the mouse, we ended up doing this front back. So we sonicated high, more towards the nose on the mouse. And that's where you would see the head and neck muscles. And then we moved the focal spot, and the circles here are the size of the focal spot. Then we moved the focal spot back, and then you could be more over the tail muscles. And in this movie, we see, and I may have shown this already, but I'll just show it again, because when it's more anterior, you see that the tail didn't move very much. And then when we move it back more posterior, in my mind, it's posterior, but it's really caudal. Now you see the tail really goes a lot. And then we'll move it back more rostral, and then you'll see that the tail movement will decrease again. So yeah, like that. So we were able to get some localization in the mouse, just not lateralization side to side. So we wanted to see, here, since we're sonicating one side, LGN, whether or not there was any lateralization. So we're able to look at the electrodes on one side versus the other side. There actually were 5 electrodes back here. So with the 5 electrodes, we could process them by looking at the left 3 and then the right 3. And then we used a sort of a double difference method where you look at the difference between 1 and 2 and 2 and 3, and then the difference between those 2 differences. And so that we're really sort of isolating the signals on that side of the brain and not looking at the long range signals. So anyway, we're looking at the 2 different sides. And here what we see is that the LGN, when we sonicated the LGN, the signal that was ipsilateral is given in blue, so it's much smaller than the signal, the visually evoked potential that was measured contralateral, which is much bigger. Now here we didn't look at the 70-100 peak to peak, but instead we looked at all of this activity out here, and we sort of looked across 250 to 450 milliseconds and looked at the RMS in order to get a metric of all of that activity. And then what we see is we look across 4 different animals in each group. So or just a total of 4 animals. And we're comparing the ipsilateral side to the contralateral side, that there was a statistically significant difference when in that second LGN sonication period, and that the ipsilateral had more suppression, had some suppression compared to the contralateral. And
so to be clear here, Kim, the ipsilateral just means that you are delivering ultrasound to the same side of the brain that you're recording, or the same fiber tract, really.
That's right.
And
And
then the
those are the recordings
contralateral,
from the same side of the brain that we're sonicating. That's right.
okay, and then
And so,
contralateral, you're stimulating one fiber tract and measuring from the other. Is that right?
so we're sonicating on this side, and then when we measure from the electrodes on the other side, then the sonication on the first side shouldn't matter.
Mm-hmm.
Yeah. Okay. And so we see the suppression of the ipsilateral, and the interesting thing is that that seems to carry over into the next control period. It wasn't statistically significant, but it definitely looks as if there's still a difference between those. And then they go back and back together again. And so that was really interesting as well. You know, again, we don't have very great temporal sampling, but it was interesting to see that, especially in light of the fact that other people are looking at the fact that this could have a lasting effect. So this is,
Sorry, Kim, the first experiment you showed was all ipsilateral. Is that right?
no, the first experiment was looking at all of the electrodes.
Oh, I see, I see. Okay, so there is some amount of crossover. Does your field crossover at all, or is the field very localized to one tract or the other?
No, they were just flashing lights for the whole visual field.
Oh, sorry, I mean the ultrasound.
No, it was on one side. It doesn't go over to the other side.
That's interesting. Yeah, I wonder why, because it looks like your first LGN stimulation doesn't show any effect either, whereas the previous experiment had a nice effect there.
Yeah. Mm-hmm.
But that's science, isn't it?
Yeah.
Unexplained sometimes.
All right, so the effect was lasting for a little while, and this is really interesting because other people in the field are using that. So here's one example of a paper by one of the speakers who's going to come later in the class, Leonard Verhagen, and his group, looking at non-human primates. And specifically what they were doing was they were sonicating in the non-human primate outside the MRI for 40 seconds. And after 40 seconds of stimulation, they then put them in the MRI and did resting state fMRI and found changes in their resting state circuit over the next hour. So for example, they're looking at differences between here, and black versus purple, a little hard to see, but they definitely were seeing differences in the resting state circuitry that lasted for an hour. So that was pretty interesting to see.
Yeah, and these colors are basically indicating that that region is connected to the other regions to that degree. So, very high coupling, very high Fisher's coupling, high temporal correlation between firing of that region and others. If I'm saying that right, I think so.
Okay, so now you know what our sheep studies are. And so let's talk a little bit about the safety. And let me just give you a prelude to that. And that is that, first of all, people have looked at the safety of different animals. And these are also, for the most part, around the FDA limits. And so this is, what people are using. And so there's every expectation that we're not going to see anything when we look on the histology. So this is one paper here by Robert de la Piazza, and this is the one that I just mentioned where they were stimulating the median nerve and then looking at the evoked potential, and you see in black, but then when they sonicated that they were able to suppress the evoked potential. So, and that effect was lasting like 10 minutes. Okay, but then they took out the brains of their pigs and looked on the H&E histology, and basically they didn't see anything. This is all just looking like normal brain on H&E histology. There was another paper by Sung-Shik Yu's group, and Sung-Shik Yu will come and talk in the class a little bit later. And they were doing something very similar where they were looking at visually evoked potentials. Here they're stimulating the visual cortex and looking at visually evoked potentials. But what they did then was they took their sheep brains, now it's the sheep model, they took the brains out, and what they reported was that in half of their sheep, so I think it was 4 out of the 8, that they found these hemorrhages. And then they also said that these were the ones that had repetitive sonications about 600 times.
And Kim, hemorrhages, you mean micro rupturing of blood vessels and blood getting into the brain? Is that right?
Yes, as in, I think so. I'm not totally clear that
That
exactly.
might not be the marker there, but that's sort of what they're looking for, right?
I mean, they see blood products. As to whether the ultrasound caused it, it caused a hemorrhage, I don't know that. I'm just saying there is blood in the tissue. Yeah.
Oh yeah, sorry, I'm just calling attention to like what is the definition of a hemorrhage for someone who doesn't know.
Okay. Yeah.
Not that it was caused by ultrasound.
Right. So, I mean, they see blood products in the tissue. And so, and they said, well, those are ones that we're seeing with repetitive sonication. So I personally felt as if this was a bit of a speed bump, and that we needed to redo the study, either corroborate it or explain it. So that was the reason why we did our sheep study. We also did this other one that was that reported in the same paper. And our methods are very similar here in that we looked at H&E histology, just like the last couple of papers did. Here we looked at two different non-human primates. They had to be euthanized because they had a disease. And we just sonicated the back of the brain. So this is around visual cortex. Using this kind of a lower frequency ultrasound. It was pulsed and 500 repetitions. Just put the transducer on, sonicated, and then estimated what the in situ pressures must have been. And then from that, we could calculate the ISPPA and the ISPTA. Now, for the ISPPA, we were within the FDA limit. For the ISPTA, we were above that limit. So in some ways, it's nice to go above the limit so that you could, really push it and say, well, we didn't see anything when we really pushed it. So therefore, if we had been at the limit, we wouldn't have seen anything then either. So what did we see? First of all, we took 3 different slices. So one that was 2 centimeters in, about where the focal spot should have been, and then 2 that were closer to the surface. And then looking at the H&E histology, we didn't find any evidence of necrosis. Necrosis would be cells breaking up. In fact, all these cells just look very, very normal. And again, with H&E histology, the darker purple are the nuclei and the lighter purple is cytoplasm. There's no red blood cells here. So there's no evidence of necrosis. We didn't see anything evidence of apoptosis that we might have seen on H&E. Didn't see any edema, no hemorrhage. I mean, we just saw in the last couple of slides what hemorrhage looks like. So there's, we can't see that here. No other evidence of inflammation or neuropil rarefaction. Okay, so really not a lot to see there. And then there's our sheep study. So a little bit different with our sheep study in that we had a number of sheep and then we really sonicated them. We had 2 different sonication schemes. One was the MRRF images that you saw. Those tended to use higher intensity with shorter duration pulses. And then the neuromodulation, which we repeated a lot with lower intensity. So I'll show you the intensity plots in just a moment. We had 4 different groups. So the first group had focus ultrasound, and then they were euthanized within an hour or so after the experiment. The second group had delayed euthanasia. So in the first group, they would, if there had been a hemorrhage and some kind of inflammatory response due to the hemorrhage, they wouldn't have shown that because that inflammatory response is optimal, it maximizes about 4 days later after the injury. And so we delayed the euthanasia for 4 days and euthanized them at that point. The third group had repeated focus ultrasound experiments, and then 4 days after their last one, then they were euthanized. And the last one was a group that had very, very high dose and repeated, and then they were euthanized 4 days later as well. Okay, so this is the data that we collected. This here shows you the pressures that we used and then the number of focus ultrasound bursts. And so compared to Sung-Shik-Yu, who had 500, we wanted to push it. And especially for the high dose cases, we really had a lot of bursts. These over here at the lower pressures were the neuromodulation sonications, and then the MRRF sonications are more over here. We always kept those locations separate, so we never did MRRFE at the same location that we did the neuromodulation. Just in case there were differences to be seen, we would be able to sort it out. In terms of the number of bursts versus ISPTA, so that's shown here, that kind of gets back to the point I made earlier, is that, pressure is more indicative of mechanical effects. ISPTA is more indicative of thermal effects. So it's kind of nice just to present both. And then in terms of the FDA limits, so the FDA limits are given above. So for MI of 1.9, so that turns out that that's this point on the graph. ISPPA of 190 watts per centimeter squared. So that's this point on the graph. So I just converted that over now to pressure. And you can see most of our sonications were below those points. That was, so we were seeing effects when we were within the FDA limit for ISPPA and MI. Some of the sonications were above those. And so that's good because it's nice to also push the limit when you're looking at safety, to go above the limits and really, really hit it really hard and say, do you see anything at that point? In terms of ISPTA, the limit is shown here, and all the sonications were above that limit. So that's good for the safety study. It just turns out that it's hard to stay within that limit because you have to have a lot of dead time between the sonications, and we didn't wait that long between the sonications. So, but in terms of safety, it's good to really, go way beyond in order to say that there wasn't anything at the very large sonication duration.
And Kim, just to confirm, even though they were separate studies, when you said neuromodulation here, you used the same visually evoked potentials metric.
Yep.
For all of those points. Okay.
Yep. Okay, so that's the data. And then this is the results. For every sonication, we took 3 slices, one right straight through it, and then 3 millimeters in front and behind. And so you can see that we evaluated a fair number of slices in every brain. Okay, so these are the different groups shown in the columns. And not every sheep had sonications on both hemispheres. So circled in green is the hemisphere that was sonicated. And then the top row is red blood cell extravasation. So we did see that in all of the sheep. This is red blood cell extravasation in the meninges. And so the meninges are kind of a layer out on the outer part of the brain. And we saw it in all of the groups that had sonications, as well as then the 2 sheep that didn't have any ultrasound. So remember, I had said that we did the MRRFE, and that was really useful because it was good to make sure the equipment was working, because there was a couple of times that it wasn't working. And when we didn't see any MRRFE sonication, we said, the equipment's not working, right? But those became our control sheep. And so it was actually nice that we were able to. What's that?
That worked out for you.
Yeah, it worked out really well. Okay, so we did see meningeal red blood cell extravasation. We saw it in all of the sheep. So that was unlikely to be due to the focus ultrasound. There was perivascular red blood cell extravasation. And again, we saw that in all of the sheep. And so again, that would not have been due to the ultrasound. We never saw red blood cell engulfment, hemosiderin-laden macrophages, or neuronal necrosis. Now, all of these would have been effects that would have shown up in that 4 days later, that would have been signs of sort of an inflammatory response to an event. And we never saw that. So if there was any red blood cell extravasation that occurred due to the extraction of the brain, then that would not have had any of the inflammatory response. And so some of this could have been due to the extraction of the brain. The fact that we didn't see any inflammatory response meant that it wasn't due to the ultrasound that we had done 4 days earlier.
So Kim, the top 2 metrics you could get in an animal that's already been euthanized, but all the bottom 3, the animal would still have to be alive with a fighting immune and repair system.
Yeah. Okay, so that's the basic result, is that we didn't see anything that wasn't also seen in the controls, and there was no evidence of inflammatory response showing that the ultrasound had done anything. The last thing that I don't have any data to show you, but we had also done some tunnel stains, and tunnel is good at showing apoptosis. Apoptosis is sort of programmed cell death. So if a cell is kind of has like a low level of injury, then it may go on to sort of die a little bit later. And so that's apoptosis. And so we had not seen any evidence of that. It's sort of less of a strong event. It's more like a, very subtle event that's gonna end up, it's just gonna like give up a little bit later. Does that kind of explain it? Or do you have a better explanation for apoptosis?
No, that was perfect.
Okay.
Or at
Anyway,
least
we
it
didn't
was
see
very
anything
good.
on tunnel stains either. So that's our safety study in sheep. And so I felt like this was really important, at least for me, to feel as if I could proceed now with thinking about human studies. I think that there's still more to do, that we have more tools available to us other than just H&E and tunnel. And I'd like to follow up with all those different tools. We can do more in terms of looking at inflammatory response and immunohistochemistry. But just, the initial thing I think is very promising, that we didn't see anything here, and it needed to be done. Okay, so some just last comments before we quit. And there's this question of which is most limiting for us. So I think I've already made that point, that the ISPTA is really the most limiting for us. And, I kind of questioned that one step further, where I said, well, if we're going to maximize the MI, then what is the ISPPA that corresponds to? So that corresponded to an ISPPA of 58 watts per centimeter squared. And then I said, well, in order to maximize ISPTA, then with that pressure, what would our duty cycle have to be? So remember that the ISPTA is equal to the duty cycle times the ISPPA, right? So what does the duty cycle have to be so that we can stay under ISPTA with that ISPPA? And it turns out the duty cycle has to be 1/80, which is a very small number. Usually we have duty cycles that are like 0.3/1. And even if we do like a burst, a series of bursts, so we have a 50% duty cycle here, so that we get that 0.5, and then we have a burst duration of 300 milliseconds and repeat the whole thing at 1 second, then that's another 0.3, and so that's 0.15, and we'd have to have an interstimulus interval of 12 seconds in order to be under that ISPTA. And 12 seconds is a really long time. So that was just sort of interesting to me, to sort of work through the numbers, how to stay under all of the limits. We'll see. At this point, I don't think anybody's really talked to FDA about what should the limits be for neuromodulation. So I think it'll be interesting to have that conversation with them.
I don't think we should mention specific cases, but we have seen people just literally go up to the limit. When they can, they just go up to it. And I don't know if that's wrong, but it is what it is at the time.
Well, the FDA limits right now, I think, are pretty low. So,
well, your paper clearly shows that they're way too low, probably.
yeah.
So I guess the question is, why wouldn't you go right up to the limit?
Right. Right.
Maybe we all should be, and maybe you should be trying to expand upon it so we get more effective treatments and still keep them safe.
Yeah. All right, well, I think that's about all we have for today. Let me just summarize. Suppression of the visual evoked potential, we stayed under ISPPA and MI limits, although we were over the ISPTA limit. The safety study in the sheep, we didn't see anything on H&E or tunnel. We were way over the FDA limit for MI and ISPTA, so that was good that we were really able to sort of push it in order to look and didn't see anything. And then what would be most limiting for us, what will be most limiting, and ISPTA looks like it'll be most limiting. So